In recent decades, the increasing awareness of environmental contamination and its cumulative impact on human physiology has led to a surge of scientific attention on the relationship between synthetic materials and biological systems, especially within the aging population. The widespread presence of plastic products across virtually all aspects of daily life has resulted in a chronic, often invisible exposure to chemical compounds that migrate from these materials into food, water, household dust, and the broader ecosystem. For older adults, whose bodies have undergone decades of interaction with such materials, the biological footprint left by plastic-related toxins becomes a subject worthy of deep academic consideration. As biochemical resilience diminishes gradually with age, the body's reduced capacity to metabolize, neutralize, and excrete foreign compounds creates a markedly different internal environment compared with that of younger individuals. Thus, the tendency of various plastic-derived substances to accumulate in the bloodstream is not merely an environmental issue, but a gerontological one, linking modern industrial chemistry directly with the physiological vulnerabilities of aging. Plastics are composed not only of polymer chains, but also of a multitude of additives that modify texture, stability, flexibility, and durability. Over time, and under specific conditions such as heat, acidity, mechanical abrasion, and prolonged storage, these additives can detach from their polymer matrices and enter consumables or the surrounding environment. Once these compounds gain entry into the human body, they circulate through the bloodstream, influencing cellular pathways and interacting with endocrine, metabolic, neurological, and immunological systems. The concern becomes more pronounced in older adults, whose detoxification functions, particularly hepatic enzyme activity, renal filtration, and mitochondrial efficiency, have naturally declined as part of the aging process. Consequently, the same level of exposure that a younger individual may tolerate or eliminate effectively might persist for far longer in an elderly person, magnifying the long-term implications of these chemical intrusions. Among the substances most frequently detected in biological samples, bisphenol A, BPA, has emerged as a key representative of endocrine-disrupting chemicals associated with plastics. BPA is known for its ability to mimic estrogenic activity, binding to hormonal receptors and altering the normal signaling processes within cells. When BPA circulates within the bloodstream of older adults, the hormonal imbalances it may provoke can become more complex than those observed in younger cohorts because the endocrine system in aging individuals is inherently more sensitive and less capable of rapid correction. Scientific studies have associated BPA exposure with perturbations in blood pressure regulation, metabolic processing, and neurocognitive stability, functions that are already susceptible to decline with age. The interaction between an age-weakened endocrine system and a persistent hormone-mimicking chemical provides valuable insight into why older adults represent a biologically distinct risk group when considering plastic-derived toxins. Phthalates, another widespread class of plastic-associated compounds, further contribute to this toxicological landscape. These agents, commonly used to increase flexibility in plastics, have been shown to migrate even in low-temperature environments and without direct heat exposure. Phthalates have been linked to disruptions in metabolic enzyme activity, alterations in lipid processing, and inflammatory responses. The presence of phthalate metabolites in the bloodstream has been correlated with respiratory difficulties, particularly in older individuals who may already exhibit reduced lung elasticity, diminished gas exchange efficiency, or underlying chronic conditions. Once internalized, these compounds interfere with cellular communication and may exacerbate inflammation-based conditions that are prevalent in older age groups, illustrating the intersection of environmental toxicology with geriatric medicine. As plastic products degrade through physical wear, exposure to ultraviolet radiation, or thermal stress, polyvinyl chloride, PVC, can release chlorinated organic compounds known for their hepatotoxic potential. The liver, responsible for filtering and detoxifying the bloodstream, is central to the processing of foreign chemical agents. However, the natural aging of hepatic tissue involves reduced enzymatic responsiveness, diminished regenerative capacity, and slower metabolic throughput. When chlorinated plastic byproducts enter the bloodstream of older individuals, they tend to linger longer, impose greater oxidative stress, and may gradually alter metabolic pathways involved in detoxification. This creates a feedback loop wherein the very organ responsible for mitigating chemical exposure becomes increasingly impaired by the cumulative impact of such substances. Thus, the biology of aging transforms what might otherwise be a mild toxic burden into a more clinically significant concern. In recent years, microplastics, defined as plastic particles less than 5 millimeters in size, have emerged at the forefront of scientific investigation, not merely as environmental pollutants, but as potential biological participants within human physiology. Their ability to penetrate membranes, diffuse through tissues, and circulate within the bloodstream challenges traditional assumptions regarding environmental exposure. Microplastics have been found in human blood samples, lung tissue, gastrointestinal lymphoid tissues, and even within the placenta, demonstrating their ability to move through biological barriers once thought to be nearly impenetrable. For older adults, whose immune and inflammatory responses are often dysregulated due to age-related changes, the presence of microplastics can induce additional stress on systems that struggle to maintain equilibrium. Scientific hypotheses propose that microplastics may contribute to chronic low-grade inflammation, oxidative stress, and immunomodulatory disruption, processes closely associated with aging and age-related diseases. Styrene, commonly released from polystyrene foam products, presents another chemical entity with neurological and hematological relevance, especially when associated with hot food or beverage exposure. Older adults may experience heightened sensitivity to chemicals that interfere with neurotransmitter regulation or cerebral blood flow, making styrene's presence in the bloodstream a potential contributor to headaches, cognitive fog, dizziness, or general neurological discomfort. The compound's volatility and ease of migration when exposed to heat amplify the risk for populations who frequently rely on foam-based food containers due to convenience or economic constraints. The biological interaction between styrene and aging neural systems remains an important area of study that illustrates the multidimensional nature of plastic toxicity. As research expands into the spectrum of toxicants associated with plastic degradation, dioxins emerge as one of the most persistently concerning categories. These compounds form primarily during the combustion or extreme heating of chlorinated plastics. And although exposure pathways differ from those of substances that leach directly from consumer products, their biological consequences are uniquely severe. Dioxins possess extraordinarily long biological half-lives due to their lipophilic nature, enabling them to accumulate in adipose tissues over the course of years or even decades. Once integrated into fat stores, they may slowly re-enter the bloodstream through metabolic turnover, thereby exerting long-term systemic effects. Older adults, who often exhibit changes in body composition, such as increased adipose tissue and altered lipid metabolism, may store and release dioxins differently from younger individuals. This altered kinetic profile amplifies the significance of even seemingly minor exposures earlier in life, suggesting that cumulative toxicological histories strongly influence present-day physiological outcomes. Another dimension of plastic-derived toxicity involves the presence of trace heavy metals, such as cadmium, lead, and mercury, historically incorporated into plastics as stabilizers, pigments, or strengthening agents. With time and material degradation, these metals may leach from plastic matrices into consumable contents or surrounding environments. Once mobilized, heavy metals enter the bloodstream and distribute themselves across neural, renal, and skeletal systems with patterns that are particularly detrimental to older individuals. Cadmium, for example, exhibits a strong affinity for renal tissue and possesses a biological half-life spanning decades, meaning that exposures from early or midlife can persist into older adulthood when renal filtration capacity has already declined. Lead, meanwhile, interferes with the mineralization process in bones and may re-enter circulation during age-related bone density loss, contributing to neurological impairments or cardiovascular dysregulation. Such dynamics reinforce the importance of considering not only present exposures, but also the accumulated chemical burdens that aging bodies have silently carried for many years. Underlying all these specific toxicological interactions is a broader physiological principle. Aging fundamentally alters how the human body responds to environmental contaminants. Declines in mitochondrial function reduce the efficiency with which cells neutralize reactive oxygen species, thereby magnifying oxidative stress generated by plastic-associated chemicals. Immunosenescence, age-related changes in immune competence, reduces the body's ability to identify and eliminate harmful foreign substances or damaged cells. Endocrine rhythms, once tightly regulated, become more fragile and susceptible to interference by exogenous hormone-mimicking compounds. Reduced renal and hepatic capacity further limit the ability to excrete toxicants promptly, making the bloodstream a longer-term reservoir for substances that would otherwise be cleared rapidly in youth. In this context, the presence of plastic-derived compounds in the bloodstream of older adults is not simply a matter of exposure, but rather the result of a complex interplay between external chemical pressures and internal physiological constraints that arise with age. Moreover, the chronic nature of exposure adds another layer of complexity. Unlike acute poisoning events in which symptoms are abrupt and unmistakable, the infiltration of plastic-derived toxins often manifests subtly through low-dose, long-term exposure patterns. Such exposures may contribute to systemic inflammation, metabolic dysfunction, or neurophysiological disturbance in ways that are difficult to attribute to a single source. This subtlety complicates epidemiological analysis and may lead individuals to underestimate the potential significance of seemingly benign daily habits, such as microwaving food in plastic containers, storing leftovers in aging plastic packaging, or consuming beverages from bottles exposed to sunlight. For older adults, whose physiological threshold for disruption are lower, these cumulative exposures may contribute in incremental yet meaningful ways to the development or progression of age-related disorders. As toxicological research progresses, it becomes increasingly evident that the interaction between plastic-derived contaminants and the aging body is not merely additive but often synergistic. For example, oxidative stress induced by microplastics or phthalates may exacerbate existing chronic inflammatory conditions such as arthritis or cardiovascular disease. Endocrine disruption from BPA may interact with age-related metabolic changes, influencing insulin sensitivity or lipid metabolism. Heavy metals that accumulate within neural tissue may compound age-associated cognitive decline, blurring the lines between environmental influence and intrinsic neurodegenerative processes. These combined effects underscore the value of a holistic scientific approach that integrates gerontology, environmental toxicology, molecular biology, and public health to more fully understand the nature of the threat. Despite these concerns, the human body retains remarkable adaptive potential. Research on geriatric health demonstrates that even in advanced age, biological systems respond positively to interventions aimed at reducing toxic exposures and supporting detoxification pathways. The liver can improve its metabolic efficiency when supported by appropriate nutrient intake. The kidneys maintain their filtering capacity more effectively under conditions of adequate hydration, and cellular repair mechanisms, including autophagy, respond robustly to lifestyle patterns that minimize oxidative stress. Reducing contact with sources of plastic-derived contaminants therefore has practical significance, not only preventing further toxic accumulation, but also allowing existing physiological pathways to gradually reduce toxic burdens already present in the bloodstream. Scientific literature also emphasizes the importance of environmental justice in discussions of plastic toxicity among older adults. Individuals of lower socioeconomic status or those living in areas with limited access to alternatives to plastic packaging may experience disproportionately higher exposure levels. Similarly, Older adults living in long-term care facilities or relying on convenience foods may unknowingly encounter elevated levels of plastic contaminants due to reliance on inexpensive or single-use plastic materials. These environmental disparities highlight the need for broader public health initiatives aimed at improving access to safer materials, raising awareness about exposure reduction strategies, and implementing policies that limit plastic use in high-risk settings, particularly those serving aging populations. In parallel with public health efforts, ongoing scientific innovation aims to develop biodegradable plastic alternatives that minimize chemical leaching and reduce environmental persistence. Yet even as promising materials emerge, understanding the full toxicological profiles of these alternatives remains an essential research priority. Past experience demonstrates that replacing one material with another does not guarantee improved safety unless the new material's additive profile, degradation products, and environmental interactions are rigorously evaluated. The study of plastic toxicity is therefore both a retrospective and forward-looking scientific endeavor, requiring awareness not only of the challenges posed by existing materials, but also of the potential unintended consequences of newly developed substitutes. The complexities of plastic-related toxicology reveal the intricate interactions between modern material science and human physiology. In older adults, whose biological systems function within narrower homeostatic ranges, these interactions acquire heightened significance. The bloodstream, as the primary transport medium for both nutrients and contaminants, becomes a reflection of lifelong environmental exposures, lifestyle patterns, and physiological changes. Understanding the nature and impact of plastic-derived toxins within this biological context is fundamental not only to advancing academic knowledge, but also to informing public health strategies that support healthier aging. Within this broader scientific context, it becomes essential to consider how exposure pathways themselves evolve across the lifespan. Younger individuals often encounter plastic contaminants through active behaviors such as frequent consumption of packaged foods, use of personal care products with plastic-derived compounds, or occupational exposure in industrial environments. Older adults, however, frequently experience exposure through passive environmental contact, accumulated bodily stores from earlier decades, and age-dependent physiological inefficiencies that prolong the retention of toxicants in circulation. The seemingly innocuous presence of plastic containers in household kitchens, plastic piping in domestic water systems, and synthetic fibers in clothing contributes to a low but persistent exposure load that gradually permeates biological barriers. Over many years, the slow but consistent introduction of plastic-associated compounds into the bloodstream creates an internal chemical landscape shaped not by acute poisoning, but by a form of chronic environmental immersion. This phenomenon raises an important scientific consideration. The threshold of toxicological relevance may be significantly lower in older adults than in younger populations. Toxicology traditionally relies on dose-response relationships, wherein biological effects are expected to increase in proportion to the amount of exposure. Yet endocrine-disrupting chemicals, such as BPA and certain phthalates, often exhibit nonlinear dose-response curves, meaning that even extremely low concentrations can exert meaningful effects on physiological systems. When combined with the diminished ability of older organs to compensate for stressors, this nonlinearity challenges traditional assumptions about safety thresholds. The bloodstream of an older adult may reflect not a single toxic event, but a constellation of low-level contaminants whose combined influence produces significant physiological consequences despite individually subtle concentrations. Microplastic exposure presents another dimension of complexity, particularly regarding its physical characteristics. Unlike chemical toxicants, microplastics may induce mechanical irritation as they interact with tissues. Emerging studies demonstrate that microplastics can cross epithelial barriers in the digestive tract, enter lymphatic circulation, and subsequently reach the bloodstream. Their surfaces are often coated with environmental pollutants, microbial biofilms, and residual additives from their original plastic forms, allowing them to act as vectors that transport multiple categories of contaminants simultaneously. Within the bloodstream, their interactions with immune cells may induce localized inflammatory responses, influence cytokine production, or alter the oxidative state of surrounding tissues. Older immune systems, which already display shifts toward chronic low-grade inflammation, a phenomenon termed inflammaging, are particularly susceptible to amplification of such responses. The addition of microplastic-induced inflammatory activity to this pre-existing baseline may contribute to physiological conditions that are difficult to detect clinically yet relevant to long-term health. The interactions between plastic-derived chemicals and metabolic processes in older adults offer another scientific frontier worthy of exploration. Age-related changes in glucose metabolism, lipid processing, and mitochondrial efficiency create physiological environments in which foreign chemicals may exert amplified effects. BPA, for instance, has been shown to influence insulin receptor signaling and adipocyte differentiation. In younger individuals, metabolic systems may compensate effectively for such disruptions, but in older adults, particularly those with pre-existing metabolic disorders, the presence of endocrine disruptors in circulation may intensify dysregulation. Meanwhile, phthalates have been associated with altered lipid profiles, influencing cholesterol transport and potentially contributing to cardiovascular risk. When such interactions are viewed against the backdrop of aged physiology, the importance of minimizing exposure becomes increasingly clear, not due to acute toxicity, but because of the subtle magnification of vulnerabilities inherent in aging. Neurological implications of plastic-derived contaminants also merit serious consideration. The aging brain undergoes structural and functional changes, with reductions in synaptic density, neurotransmitter concentrations, and neuronal plasticity. Chemical agents, such as styrene or heavy metals, have the potential to interact with these vulnerable neural pathways. Styrene, for example, may affect dopaminergic signaling or cerebral blood flow dynamics, creating cognitive disturbances that could easily be mistaken for ordinary aging. Heavy metals, once accumulated in neural tissues, may disrupt calcium signaling, impair mitochondrial function, or contribute to the aggregation of misfolded proteins. These subtle neurotoxic effects may cumulatively accelerate age-associated cognitive decline, revealing an environmental component to processes often attributed solely to intrinsic aging. The presence of such contaminants in the bloodstream suggests that environmental exposures decades earlier may intersect with the neurological trajectories of older adults in ways that science is only beginning to uncover. From a cardiovascular perspective, plastic-derived contaminants present additional complexities. Endocrine disruptors may influence vascular tension, modulate nitric oxide production, or interact with hormonal pathways that regulate blood pressure. Microplastics may contribute to endothelial irritation or influence coagulation dynamics. Persistent organopollutants like dioxins can alter lipid metabolism and induce oxidative stress that affects vascular integrity. When combined with the natural stiffening of arteries, reduced baroreceptor sensitivity, and increased oxidative load associated with aging, these chemical influences may contribute to cardiovascular instability in ways that are subtle yet clinically meaningful. Understanding how chronic exposure intersects with age-related cardiovascular changes remains an important area for interdisciplinary research that bridges environmental science, cardiology, and gerontology. Furthermore, the biological half-lives of many plastic-derived contaminants intersect critically with the temporal dimension of human aging. For example, cadmium's half-life of several decades implies that individuals exposed during middle age may continue to experience its effects well into late adulthood, even if external exposure has declined significantly. Similarly, lipophilic toxicants such as dioxins, which accumulate in fatty tissues, may re-enter circulation during periods of weight loss, common in older adults due to illness, reduced appetite, or metabolic changes. This dynamic creates a paradox wherein older adults may become more toxicologically burdened precisely when their bodies appear to be eliminating older fat stores. Understanding this interplay between aging metabolism and stored toxicant release is crucial for comprehending the full scope of plastic-derived biological influence. Scientific evidence also suggests that the interactions between multiple contaminants can yield outcomes that are not predictable from studying each substance in isolation. Plastic-associated chemicals often share endocrine-disrupting properties, generate oxidative stress, or influence inflammatory pathways. When present simultaneously, as is common in real-world exposure scenarios, their combined effects may be synergistic rather than simply additive. Older adults, whose physiological buffers are already diminished, represent a population in whom synergistic toxicological interactions may be particularly consequential. This emphasizes the importance of studying not merely individual contaminants, but the combined chemical environments that accumulate in the bloodstream across a lifetime. The emerging understanding of these synergistic interactions calls for a more nuanced approach to evaluating toxicological risk in older populations. Traditional toxicology often relies on controlled laboratory conditions in which single compounds are administered in isolation, enabling researchers to map dose-response relationships and determine specific biochemical pathways of disruption. However, the real world rarely conforms to such simplified scenarios. Older adults are continually exposed to complex mixtures of contaminants originating from plastics, environmental pollution, dietary sources, pharmaceuticals, and metabolic byproducts inherent to aging itself. Within this composite chemical environment, the presence of plastic-derived toxins represents just one component of a broader ecological network of exposures. The bloodstream becomes a biochemical archive in which residues of lifelong encounters with synthetic materials coexist with endogenous compounds, creating dynamic interactions that require interdisciplinary scientific frameworks for full comprehension. As researchers investigate these cumulative effects, increasing attention is being directed toward the concept of exposomics, the comprehensive analysis of all environmental exposures across an individual's lifetime. When applied to older adults, exposomics reveals that the physiological signatures of plastic-derived contaminants cannot be separated from the broader context of aging-related biochemical shifts. For example, lower glutathione levels, characteristic of older individuals, diminish the body's ability to neutralize reactive oxygen species generated in response to chemical influences. Mitochondrial DNA mutations, which accumulate naturally with age, further impair the cell's capacity to endure oxidative challenges. The introduction of plastic-derived toxicants into this already strained system intensifies the biochemical turbulence in ways that may not be immediate or overt, yet remain biologically significant. Environmental toxicology also intersects meaningfully with sociological and behavioral variables, further shaping exposure patterns in older age. As mobility decreases and dependence on processed or convenience foods increases due to reduced physical capacity for meal preparation, older adults may inadvertently consume greater quantities of food stored in plastic packaging. Similarly, the increased use of medications, many of which are stored in plastic blister packs or bottles, may contribute indirectly to plastic-related exposure through handling or contamination pathways. Aging-in-place practices, common in many societies, often involve the long-term use of household items such as plastic bowls, cups, utensils, and storage containers that have undergone years of thermal cycling, chemical wear, and ultraviolet exposure. Over long periods, the degradation of these items may result in elevated release of contaminants, such that older adults are uniquely positioned to receive a disproportionate share of household plastic leachates simply because they have lived with these objects for longer durations. The phenomenon of cumulative exposure is not only biological, but also temporal. Older adults bear the historical legacy of earlier eras in which plastic regulation was minimal or non-existent. Decades ago, plastic products were manufactured with little consideration for long-term leaching behavior or human exposure potential. Additives such as phthalates or heavy metals were employed freely, and consumer awareness regarding chemical migration was limited. Thus, individuals who are now in their 60s, 70s, or 80s may have experienced peak exposure during periods when plastics were ubiquitous yet poorly regulated. Their current bloodstream compositions may therefore reflect both contemporary exposures and residual contaminants accumulated during earlier stages of life. This temporal dimension underscores the value of longitudinal analysis when examining plastic toxicity in aging populations. Within the internal environment of the aging body, the consequences of plastic-derived contaminants unfold not as abrupt biological crises, but as subtle modifications to homeostatic balance. Homeostasis, the body's ability to maintain stable internal conditions, gradually becomes more fragile with age. Small perturbations can trigger cascading effects across biochemical networks. For example, endocrine disruptors such as BPA may alter hormonal feedback mechanisms that regulate blood glucose, lipid metabolism, or vascular tone. These disruptions may appear minor in isolation, but can exert disproportionate influence within an endocrine system already contending with age-related hormonal variability. Likewise, microplastics generating localized inflammatory responses may contribute to a cumulative inflammatory burden that exacerbates conditions like atherosclerosis, insulin resistance, or neuroinflammation, each of which is more prevalent among older adults. From a molecular perspective, oxidative stress emerges as a central mechanism through which many plastic-derived contaminants exert their influence. Reactive oxygen species generated in response to chemical irritation, foreign materials, or metabolic interference can damage proteins, lipids, and nucleic acids. Younger bodies often compensate through robust antioxidant systems and efficient DNA repair pathways. However, in older adults, the capacity for oxidative mitigation declines, allowing damage to accumulate. Mitochondrial dysfunction, already commonplace in the aging process, further amplifies vulnerability to oxidative insults. The introduction of toxins, such as dioxins or heavy metals, into this compromised environment intensifies cellular stress responses, potentially accelerating tissue degeneration or heightening susceptibility to chronic disease. In addition to oxidative stress, plastic-derived contaminants influence epigenetic regulation, a domain of particular interest in aging research. Epigenetic modifications, such as DNA methylation or histone acetylation, govern gene expression without altering the underlying genetic code. These modifications accumulate across the lifespan and are shaped by environmental exposures. Endocrine-disrupting chemicals have been shown to alter epigenetic patterns, potentially influencing metabolic pathways, immune responses, or cellular aging processes. In older adults, whose epigenetic landscapes already reflect decades of environmental interactions, the addition of plastic-related contaminants may trigger gene expression shifts with long-term biological impact. Such shifts may influence susceptibility to age-associated diseases, diminish stress resilience, or alter inflammatory set points. Another aspect of aging physiology relevant to plastic toxicity involves changes in barrier integrity. Biological barriers, including the gastrointestinal lining, pulmonary epithelium, and blood-brain barrier, naturally weaken with age. Increased permeability allows greater translocation of foreign substances into circulation. Microplastics, nanoparticles, and certain chemical additives may traverse these compromised barriers more readily, contributing to elevated bloodstream levels in older adults. For instance, gastrointestinal barrier permeability, commonly observed in older individuals, facilitates the absorption of microplastic particles that would otherwise be eliminated through digestion. Similarly, age-related changes in pulmonary function can enable inhaled microplastics or chemical vapors to reach deeper regions of the respiratory tract, increasing the likelihood of systemic entry. Within the immune system, age-related declines in both innate and adaptive immunity intersect with plastic toxicity in complex ways. Older adults exhibit reduced ability to neutralize foreign materials and diminished capacity to clear damaged cells. When plastic-derived contaminants induce inflammatory or immunological responses, the body may become trapped in cycles of low-grade inflammation without achieving effective resolution. Chronic activation of immune pathways, triggered by microplastics, endocrine disruptors, or chemical irritants, contributes to systemic inflammation that weakens overall physiological resilience. This interplay suggests that immune modulation is a critical dimension in understanding the long-term effects of plastic toxicity on older populations. The interaction between plastic-derived contaminants and the aging immune system also raises considerations about the potential influence of these substances on immunosenescence, the gradual deterioration of immune function associated with aging. Immunosenescence involves both reduced immune responsiveness and increased chronic inflammation, forming a paradoxical state in which the body is simultaneously less capable of fighting new threats and more likely to generate prolonged inflammatory reactions. Plastic-derived toxicants, particularly microplastics and endocrine-disrupting chemicals, may interact with this weakened immune landscape in ways that promote the persistence of low-grade inflammation, also known as inflammaging. This phenomenon is characterized by elevated circulating cytokines, altered macrophage function, and impaired resolution of inflammatory episodes. The presence of persistent pollutants in the bloodstream can heighten baseline inflammatory tone, creating additional strain on physiological systems that are already functioning with reduced efficiency. In this sense, the presence of plastic-derived chemicals does not merely challenge immune defense, but may influence the trajectory of age-related immune decline itself. The gastrointestinal system offers another window through which to assess the significance of plastic contamination in older adults. Age-related changes in digestive function, such as reduced gastric acidity, slower gastrointestinal motility, and altered microbiome composition, intersect directly with the introduction of plastic-associated contaminants into the food chain. Microplastics, when ingested, may physically interact with intestinal mucosa, causing irritation or facilitating the absorption of co-associated chemical compounds. Endocrine disruptors leached from plastic packaging may also influence microbiome composition, altering microbial diversity in ways that impact nutrient absorption, immune modulation, and metabolic regulation. The gut microbiome undergoes significant shifts with age, often displaying reduced resilience and lower microbial diversity, making it more sensitive to environmental influences. Plastic-derived contaminants may therefore shape microbial dynamics in older adults more profoundly than in younger populations, influencing metabolic health, inflammation, and even cognitive function through the gut-brain axis. Interdisciplinary research increasingly emphasizes the role of the gut-brain axis in mediating the impact of environmental toxicants on neurological health. The presence of plastic-derived chemicals in the bloodstream, when combined with age-related reductions in blood-brain barrier integrity, creates a plausible pathway through which these contaminants may influence neurocognitive outcomes. Studies indicate that certain chemicals used in plastic production can traverse the weakened blood-brain barrier with greater ease in older individuals, potentially altering neurotransmitter activity, synaptic plasticity, or neuroinflammatory states. Microplastics, once internalized, may release additives or absorb endogenous molecules that modulate their neurotoxic potential. The resulting alterations in neuronal signaling pathways could exacerbate symptoms associated with age-related cognitive decline, including memory lapses, reduced concentration, and slowed information processing. Although the precise mechanisms remain under investigation, the convergence of environmental toxicology and neurogerontology highlights the importance of understanding how chronic exposure to plastic-associated contaminants may contribute to neurodegenerative trajectories. The cardiovascular system further reflects the multifaceted influence of plastic-derived contaminants in aging physiology. Endocrine-disrupting chemicals, such as BPA, may alter hormonal regulation of vascular tension, contributing to fluctuations in blood pressure. Microplastics circulating within the bloodstream may interact with endothelial surfaces, creating localized irritation that influences clotting pathways or vascular inflammation. Dioxins and other persistent organic pollutants may affect lipid metabolism, in ways that accelerate atherosclerotic plaque formation. These interactions occur within an aging cardiovascular system already characterized by reduced elasticity, heightened oxidative stress, and diminished adaptive capacity. As a result, even modest influences from environmental contaminants may tip the balance toward physiological instability. This dynamic underscores the necessity of incorporating environmental exposure analysis into cardiovascular risk assessments for older adults. Renal physiology, too, plays a critical role in understanding the persistence and toxicity of plastic-derived contaminants within the bloodstream. The kidneys serve as the primary filtration system for removing metabolic waste and xenobiotic substances, yet renal function naturally declines with age due to reduced nephron number, diminished glomerular filtration rate, and structural alterations in renal vasculature. In older adults, these impairments prolong the retention time of plastic-derived toxicants within circulation, allowing them greater opportunity to interact with cellular systems. Heavy metals, such as cadmium or lead, exhibit particular affinity for renal tissue, causing further damage to kidney function and establishing a cycle of declining filtration and increasing toxic burden. Even low-level exposures, when combined with age-related renal decline, may produce toxicokinetic profiles that differ significantly from those observed in younger individuals. This relationship between environmental exposure and renal aging exemplifies how physiological changes transform the boundaries of biological tolerance. The endocrine system, which orchestrates numerous homeostatic processes, represents another critical intersection point. With age, endocrine glands exhibit diminished hormone secretion, altered feedback sensitivity, and changes in receptor density. These alterations create an environment in which external endocrine disruptors, such as BPA, phthalates, and certain plastic additives, can exert outsized influence. For instance, small variations in estrogenic or androgenic signaling caused by plastic-derived contaminants may have disproportionately large effects on metabolic regulation, bone density, or neurological function in older adults. The interplay between endocrine disruptors and menopausal or andropausal hormonal changes adds further complexity, as age-related endocrine fluctuations create windows of heightened vulnerability to external hormonal interference. In addition to internal physiological processes, lifestyle changes that accompany aging may unintentionally increase exposure to plastic-derived contaminants. Older adults often rely on pre-packaged foods for convenience or due to mobility constraints. Meals delivered through social services, long-term care facilities, or food assistance programs commonly involve plastic-based packaging, reheating trays, and storage containers. When such materials are exposed to heat, whether through microwaving, steam tables, or thermal insulation, chemical leaching becomes more pronounced. Furthermore, older adults may be more likely to use medications stored in plastic blister packs or to consume bottled beverages due to reduced access to fresh water sources. These seemingly routine aspects of daily life contribute cumulatively to the overall toxic burden reflected in the bloodstream. The psychosocial component of plastic exposure also warrants consideration. Older adults often maintain long-term attachment to household items accumulated across decades, including plastic containers, utensils, and storage vessels that have undergone significant wear. Items with sentimental or economic value may be used far beyond their intended lifespan, increasing opportunities for chemical leaching as materials degrade. In addition, diminished sensory perception, such as reduced ability to detect odors or visual signs of material deterioration, may further limit awareness of potential contamination sources. Understanding these behavioral dimensions is essential for designing effective public health interventions targeted at exposure reduction in aging populations. From a broader public health perspective, the issue of plastic-derived contaminants in older adults exemplifies the interconnectedness of environmental policy, consumer behavior, industrial practices, and individual health outcomes. Regulatory frameworks governing plastic production and chemical additives have evolved unevenly across regions, resulting in significant geographic disparities in exposure risk. Older individuals living in countries with minimal regulations or limited access to safer material alternatives may face heightened exposure burdens. Conversely, populations in regions with stricter regulations may still experience legacy exposure from older plastics manufactured before contemporary safeguards were implemented. These global disparities highlight the importance of coordinated international efforts to address the long-term consequences of plastic proliferation on vulnerable populations. The scientific exploration of plastic-derived contaminants in the aging population ultimately converges upon a broader question concerning modern environmental adaptation. How does the human body, shaped through millennia of evolution within natural ecosystems, respond to a world increasingly permeated by synthetic materials? The bloodstream of an older adult can be conceptualized not only as a biological medium, but as a chronicle of environmental history, retaining molecular traces of interactions with plastics, pollutants, and chemical agents that were largely absent from the human environment prior to the 20th century. This chronicling process, subtle, gradual, and unavoidable, offers a unique perspective on the cumulative biological consequences of living in an era defined by industrial materials. The long-term presence of plastic-associated toxicants in circulation reflects the complex interplay between environmental exposures, age-related physiological decline, and the body's diminishing capacity for detoxification, creating an internal chemical landscape that would have been inconceivable in prior generations. As researchers deepen their understanding of this landscape, one key insight emerges. Plastic-derived toxins cannot be examined in isolation from the broader tapestry of aging biology. For example, neurodegenerative diseases such as Alzheimer's and Parkinson's may be influenced in part by chronic inflammatory states, oxidative stress, and the accumulation of environmental toxicants, all of which are processes implicated in plastic exposure. While plastic-derived contaminants are unlikely to serve as singular causal agents, their presence within the physiological environment of older adults may contribute to a multifactorial burden that shapes the onset or progression of such conditions. Similarly, age-related metabolic disorders, such as type 2 diabetes or dyslipidemia, may be exacerbated by endocrine disruptors that influence insulin sensitivity or lipid metabolism. This does not imply that plastic contamination is the primary driver of these diseases, but suggests that plastic exposure represents a biologically relevant component within a multifaceted network of environmental and genetic influences. The cumulative nature of plastic exposure also raises considerations about intergenerational health. Older adults represent the first generations to have lived their entire lives in a plastic-rich environment, meaning that their physiological responses to these materials may offer early indications of how lifelong exposure shapes health trajectories across the lifespan. The presence of microplastics in human placental tissue, for instance, indicates that exposure begins even before birth. However, the long-term implications of such early exposures cannot be fully understood without considering the biological patterns emerging in older adults who have undergone decades of cumulative contact. In this sense, the aging population serves as a sentinel group, revealing the potential outcomes of exposure patterns that will become increasingly common in future generations unless meaningful environmental changes are enacted. Despite the extensive evidence documenting the presence and potential effects of plastic-derived contaminants in older adults, it is important to recognize that the body retains substantial adaptive capacity. Biological systems continuously adjust to maintain internal balance, employing mechanisms such as metabolic detoxification, antioxidant defenses, immune surveillance, and cellular repair pathways. Even in older age, these systems can be strengthened through targeted lifestyle modifications. Dietary choices rich in antioxidants may support the neutralization of reactive oxygen species generated in response to chemical irritants. Adequate hydration supports renal filtration, facilitating the elimination of contaminants. Regular physical activity enhances circulation, promotes detoxification processes, and reduces inflammation. Minimizing the use of plastic containers for food storage, avoiding the heating of plastic materials, and opting for glass, ceramic, or stainless steel alternatives can significantly reduce exposure to leach chemicals. These strategies, while simple, illustrate the practical potential of integrating environmental toxicology insights into everyday life to support healthy aging. The scientific community also plays a pivotal role in shaping policies and guidelines that protect vulnerable populations, including older adults. Research on plastic toxicology continues to evolve, providing new data that informs regulatory decisions regarding permissible chemical additives, manufacturing standards, and waste management practices. Environmental legislation aimed at reducing plastic pollution, such as restrictions on single-use plastics or bans on specific additives, can indirectly improve public health by limiting collective exposure. Yet policy interventions alone are insufficient without public awareness, which empowers individuals to make informed choices about their personal exposure levels. Educational initiatives tailored to older adults, emphasizing both the risks and practical mitigation strategies, can bridge the gap between scientific knowledge and daily behavior. On a broader societal level, the issue of plastic-derived contamination highlights the interdependence between environmental health and human health. The accumulation of plastic waste in oceans, soil, and freshwater ecosystems ultimately influences the quality of the air we breathe, the water we drink, and the food we consume. Microplastics ingested by marine organisms can move through food chains, eventually reaching human consumers. Chemical additives used in agriculture, industry, and consumer products may accumulate in environmental reservoirs, creating diffuse exposure pathways that affect entire populations. Older adults, with their reduced physiological resilience and prolonged exposure histories, serve as a reminder of the human cost of environmental degradation. Their bloodstream becomes a biochemical testament to the consequences of global plastic dependence, urging society to reconsider its relationship with synthetic materials. Moving forward, interdisciplinary research will be essential to deepen our understanding of plastic toxicology in aging populations. Advances in analytical chemistry may allow for more precise identification of contaminant profiles within the bloodstream. Innovations in cellular biology and molecular imaging may reveal how microplastics interact with tissues at the microscopic level. Genomic and epigenomic studies may illuminate how long-term exposure influences gene expression, accelerating or modifying age-related biological pathways. As data accumulates, a more comprehensive picture will emerge, one that situates plastic exposure within the larger context of environmental health, social behavior, and human longevity. In conclusion, the presence of plastic-derived contaminants in the bloodstream of older adults represents a complex interplay between environmental exposure, physiological aging, and cumulative toxicological burden. The biological consequences of this phenomenon are multifaceted, influencing endocrine function, metabolic regulation, immune response, neurological integrity, and cardiovascular stability. While modern life makes complete avoidance of plastic exposure unrealistic, a deeper scientific understanding of its impacts enables more informed decisions, both at the personal and policy levels. As the first generations to experience lifelong exposure to synthetic materials, older adults provide essential insights into how these exposures shape human health across the lifespan. Their physiological experiences underscore the need for continued research, environmental reform, and public health initiatives aimed at reducing harmful exposures while supporting the body's inherent capacity for resilience. Ultimately, the study of plastic-derived toxins in aging populations is not merely an examination of contaminants, but a reflection of humanity's evolving relationship with its environment, and a call to action to forge a future in which technological advancement and biological well-being coexist more harmoniously.